get started with the next talk. Our speaker is Nina from Cornell University, and take it away.
Yeah, thank you. So I'm going to be talking about some work we've been doing to explore how the access to increasingly cheaper and cheaper DNA sequencing can really substantially improve our ability to tell where fish originates from. Before I start, I just want to acknowledge that most of this work was led by my former postdoc, Franckowiak, and in collaboration, key contributions from many other people, including Eric Anderson, formerly at NOAA, Dave Kasiak, and Miluska Hyde at USGS, and our wonderful colleagues at the Hudson River Estuary Program, and many others. But genetic assignment, as I said, is really about, for a given sample, figuring out which of a series of possible populations is this individual most likely to have originated from. And this has really broad applications in ecology and conservation, especially in cases where animals move away from their breeding grounds. Then doing genetic assignment can help us get a better handle on patterns and rates of dispersal at different times of year, at different life stages, and so on, and connectivity across space. So that's used, for example, to infer what overwintering grounds are used by specific populations of migrating birds, or it can be individual level, identifying long distance dispersers or straying individuals in bear populations, or what's probably top of mind for everyone in the room here is one of the most common applications, which is to fisheries. So looking at, especially in a mixed stock situation, being able to estimate the relative abundance of different contributing stocks, so we can assess fishing impacts on each of those stocks. And one species for which there's been a lot of interest in developing genetic assignment is American shad. So as you probably know, American shad are natively distributed all along the East Coast, and it used to be extremely abundant, supporting some of the biggest fisheries, so much so that it's been called the founding fish because of the crucial role it played in sustaining early European settlers here. Unfortunately now, populations have been decimated across much of the range. And so shad are anadromous, breed in rivers, but spend most of their adult life at sea. And there have been a lot of in-river restoration efforts, but it actually looks like one of the big threats is getting caught as bycatch at sea during the marine life stage. So there's been a lot of interest in then being able to have a tool out at sea. A shad just looks like a shad, but there's been interest in figuring out where do fish from the most vulnerable river populations get caught, what times of year and what fisheries do they get caught at sea, so we can put in place some more targeted protection measures instead of just blanket closures, because some shad populations are actually doing much better than others. Unfortunately, earlier efforts to develop genetic assignments were met with some mixed success. So a really impressive study led by Dan Hasselman and colleagues published in 2013 had genotyped shad from 33 different populations. And the result I pulled out here is the result of a genetic clustering analysis where each of these tiny little lines here represent an individual fish colored by its inferred genetic ancestry. So you can see that there's this very clear clustering into distinct groups here, which looks like great foundations for assignment. But unfortunately, most of this fine-scale structure is found up in Canada. In US waters, which is where the management interest is most intense, these data only resolve two major groupings. So in a more recent study where a different group was using a similar set of microsatellites, they were also able to assign with high confidence to those two groups in US waters. But when they were looking at their assignment success to individual rivers, so here we're looking at the proportion of fish that were genetically assigned to the river in which it was caught, we see that the accuracy is disappointingly low, on average about 80%, ranging all the way down to 4%, which really limits the usefulness of this tool to assign to the level of individual rivers, which is important in this context. But in the meantime, as you probably know, there's been this dramatic technological development, and you might have seen something like this plot before that shows how the cost of DNA sequencing has dropped over the past few decades. And I just want to alert you to that this is a logarithmic axis here. Oops, so this means that there's been, a greater than 500,000-fold reduction just since the turn of the century. So that's, of course, a complete game changer in terms of what we are able to do. But even though sequencing is becoming so cheap, genomes are really huge. So we still need to be strategic about how, if we want to look at hundreds of individuals, we still have to be strategic about how we make this technology affordable enough to be applied on a broad scale, especially for conservation projects where budgets are often limited. So the traditional approach out of necessity that has most commonly been taken is to, instead of sequence the entire genome, we focus in on a small number of sites in the genome that we can then sequence and genotype really well. And that can give us a lot of insight, but what's becoming increasingly clear is that for some, when we have lake populations, so some small populations that have been isolated for a long time, we see large genetic difference across the entire genome. Those are easy to distinguish genetically. But for types of species that have either very large populations or have a lot of connectivity, often a lot of the genome won't show very much differentiation, but it'll be very localized positions in the genome. And with this kind of approach, even when we are now up to getting several thousand markers, we may often be missing some of these peaks if our sampling of the genome is not very dense. So in my lab, over the past decade, we've been really interested in figuring out ways that we can sort of spread our sequencing over the entire genome to be able to screen across the whole genome. The downside of that is then that we sequence so shallowly that we can't confidently call genotypes at any one position. But we can quantify our uncertainty quite accurately, and as long as we take that uncertainty explicitly into account and model it into our downstream inference, this is a powerful approach for making population level and individual level inference. So we applied this to the shed. We got samples from working with lots of people who helped us get fish across these 12 locations, about 30 individuals in each. And we did that low coverage whole genome sequencing. And here we're looking at a principal components analysis of just the overall genome-wide results. Each dot here is an individual, color-coded according to its sampling location. And in this PCA here, dots that are closer together are more genetically similar. So we were really happy to see that there's clear geographic patterns in the overall genetic pattern here. And in particular, we were thrilled that we could see within those two major regions that had been the resolution of what could be resolved with the microsatellites, we actually saw separation of several of the rivers. So much greater resolution. But this here is just the genome-wide pattern. So as I was talking about before, it's becoming increasingly clear that the level of differentiation between populations vary a lot across the genome. So these three panels here are showing pairwise comparisons between two neighboring populations. Along the y-axis, we have FST, which is a measure of how different allele frequencies are in those two. So what's the proportion of A in one population and the proportion of G, or the proportion of G in the different population. So a high value means there's a big allele frequency difference. A low value means they basically look the same. And then each of these tiny dots here is a position in the genome, color-coded, they're organized along the assembled genome, color-coded by the different chromosomes in the genome. And what we can see is that there's a lot of heterogeneity, and we have these, even though the vast majority of points are down here showing very low levels of differentiation, we have these regions where we see almost fixed differences. Basically, in one populations we see only A's, in the other populations we see only G's. So our initial plan was then, if we just pick some markers from those peaks, that'll really give us a lot more information, right? So we can kind of think of it, if we pick a marker from up here, top of a peak, the situation is something like this, where we have really big differences between two populations, as opposed to just any random marker, where there's much less differentiation between the two populations that we want to distinguish between. And this is before we had the tools to be able to screen across the entire genome by just sampling randomly. We were much more likely to get this scenario here. So it's exciting that we now can go in with this pre-screening and in a more targeted way get closer to that scenario here. So we did a bunch of simulations and estimated that if we just took 100 of these markers, we should be able to assign with greater than 90% accuracy to each of the rivers. So we went ahead and developed, because the sequencing the whole genome, it's still, at least at the time we started this work, seemed a lot too costly and too cumbersome to do for each fish. So we were going to develop this smaller panel that we could use. Unfortunately, we were met with a bunch of technical difficulties I won't bore you with. Many months later, we started screening almost 600 SNPs or markers. We ended up with just over 107. We're going back now and redoing this with a new set, almost done with a new panel that has about 200 and a bit more power. But I'll show you the results from our first effort here. When we, based on those 100 successfully genotyped markers, we were able to we were a little bit disappointed in the results here. You see this is showing the proportion of correct genetic assignment to these different populations, which throughout are better than what we could do with microsatellites, but we're still sort of down in the 60% range for some. When we group them in, group some of the least differentiated, we get up to more like above 75% accuracy for all, which we could work with, but I think we can actually do much better with our next generation panel that's almost ready. But in general, just sort of the headaches of getting this panel optimized really got us thinking about sort of the downsides to doing that work. It's quite costly to develop. It takes a lot of time and effort to sequence over again and optimize, and also it sort of by nature requires that we predefine a set of markers that we want to look at. Those we selected to be good at distinguishing the particular populations we had data for now. So if next year we want to assign to a different population that wasn't included in this ascertainment set, this panel might not work very well for that. So we're kind of locked into an initial set that we invest a lot in developing, and then the future applicability has some limitations. But I think the really big eye-opener was when we looked at a PCA with those 107 high information markers, and we still see that there's very good matching of geographic structure to the genetic structure we see in a principal component. But the resolution is so much less than what we saw with the whole genome data, right? So it just seems frustrating to settle for this when we could have this. So the reason why we didn't go there in the first place was that when we started out this work, basically there's a whole toolbox, lots of software to do genetic assignment, but they all require called genotype. We didn't really have the tool to work directly with this low coverage data. But fortunately, colleagues Matt DeSix and Eric Anderson in the meantime developed the algorithms and the software to be able to use these low coverage data where we can incorporate the uncertainty about genotype calls directly into assignment testing. So we implemented that, boosted our sequencing depth just a little bit. And then now when we're looking at the assignment accuracy, so here in columns are where fish originated from, in rows are where they got assigned to, and then we have the counts here. And we can see just this clustering along the diagonal, suggesting that we're getting really accurate assignments. At first, there was a little bit of assignment to neighboring rivers, but once we standardized, there were some differences in the sample sizes. Once we standardized the sample sizes, that really cleaned it up, and we're getting pretty close to perfect assignment to individual rivers here, which we're really excited about. So the question though is, how practical is this, and can we really sequence the whole genome of every fish that we want to assign? And the good news is that we actually don't have to. As long as we have the reference data across the whole genome, we actually need pretty little data from each fish that we want to assign. We don't need to match all the 1 billion base pairs in the shared genome. We just need to match maybe a few 100,000. So actually, our testing suggests that we only need about 10 cents worth of sequencing per fish to be able to assign it with this accuracy. So this basically brings this whole genome screening down to the sort of the same routine processing cost. There's some sample processing, it costs about $7 to prepare the sample, but the sequencing itself is very cheap if we do it at big scale, to sort of bunch it in. Right? So I'm almost out of time, but just sort of I just think it's still kind of early days, but I think this is really promising as potentially an alternative approach for assignment moving forward. Not saying it's ideal in every case. It has some big advantages in terms of, I think primarily that we are not pre-selecting any sets, and there's not much initial optimization involved, which makes it really flexible for future diverse applications. The downside is that it does require some more computational resources, a little bit more bioinformatic expertise to be able to run. And it's also, the method is still emerging, so there will be some validation for each particular system required. But so in some cases, doing a genotyping panel is going to be a better option, but especially these kind of species that genome-wide have very low differentiation, I think this can be really exciting. So we are doing some follow-up work in striped bass and Atlantic cod to also explore the potential of how much better it can do than genotyping panels. So with that, I'll just thank you, and if there's time, I'll take any questions.
Have you tried to, it's pretty cool, have you tried to combine any of this work with other means of, classifying fish to rivers, like morphology, anything like that, or look chemistry or shape?
Yeah, no, it would be very interesting, because this, of course, only tells us about the genetics, right? It doesn't tell us about where a fish was at a different time of its life, so doing some coupling this. It's kind of a different type of information.
Yeah, but I actually think that, I was part of a paper that came out last year about otolith shape for American shad. I'll give you the paper.
Yeah.
But it was, and I honestly, I provided fish, I did not